My name's Barber Janer, and I'm part of the Laboratory for Neurobiology and Gene Therapy, supervised by Professor Veerle Baekelandt here at KU Leuven in Belgium. And I'm really thankful, and I wanted to extend my thanks to LabRoots to allow me to attend this neuroscience event series. Let's get into it. My talk today is entitled AI-Powered Insights into Brain Pathology and Neuroinflammation, Enhancing Precision and Speed of Histopathological Analysis for Preclinical Research. The field of AI may seem daunting to a lot of neuroscientists, but nowadays it's no longer so complicated to implement AI-driven tools into our research. In this talk, I will show you how we trained multiple AI models to automate immunohistological analysis in the context of preclinical Parkinson's disease research. But first, let's have a look at the second most common neurodegenerative disease in the world and the first most common movement disorder from a histopathological perspective. The reason why Parkinson's disease patients suffer motor dysfunction is because they present neurodegeneration in multiple brain regions that control movement. Particularly, the dopaminergic pathway is severely affected. In the top image, you can see a PET scan from a control or healthy individual and a PD patient. And what is tagged is actually the dopaminergic terminals in the putamen or striatum. And I hope you can note that for the PD patient, there is less signal detected because PD patients lose these dopaminergic terminals. And actually, these dopaminergic terminals connect to the dopaminergic neuron cell bodies, which are located in the substantia nigra, which is what you see in the bottom panels. This is unstained fresh tissue from, again, a healthy individual in the left and a PD patient in the right. And I hope you can know that there are in the tissue, in the control tissue, two dark lines, kind of like forming a diagonal. And these two dark lines are the substantia nigra pars compacta. They are visible to the eye because the dopaminergic neurons, when they are healthy and they are there, and they can be seen as dark because they contain neuromelanin. However, for PD patients that lose dopaminergic neurons, you can see how basically these two dark lines have been lost. Another main hallmark of Parkinson's disease is the finding of aggregated alpha-synuclein protein inclusions inside of neurons. And PD patients also present aberrant and chronic neuroinflammation, which is characterized by the recruitment of reactive microglia to regions with neurodegeneration and alpha-synuclein pathology. Still to this day, postmortem immunohistological assessment of patients' brains is needed to make a final diagnosis in some cases, but also for research purposes. However, as the availability of human tissue is limited, our group specializes in developing in vivo models to study some of these neuropathological features and test therapeutics. A common strategy that we and many others employ is to overexpress alpha-synuclein protein through viral vector technology. We usually inject viral vectors unilaterally in the substantia nigra to mainly target the dopaminergic pathway. And this also allows us to use the non-injected side or contralateral side as an internal control. We often use neuron-specific viral vectors to selectively express alpha-synuclein in neurons, which eventually induces the formation of inclusions in connected brain regions to our injection point, the substantia nigra, but also leads to neurodegeneration and neuroinflammation. We usually assess neurodegeneration of the dopaminergic pathway by staining for the tyrosine hydroxylase enzyme, which is a typical marker of dopaminergic neurons. Hopefully you can see in the image how in the injected or ipsilateral side of these two sections from a mouse brain, there's a fainter gray intensity of the staining in the injected side than in the contralateral side. So there has been a loss of these dopaminergic neurons and dopaminergic terminals in the striatum caused by the injection of the vector. So to characterize our animal models, we routinely perform immunohistological analysis. We stain for typical markers used in PD research, like the TH marker I mentioned before. And so after staining and microscopy, we perform image analysis using different methods depending on the marker. However, this usually involves manual annotations of the anatomical regions of interest and sometimes even manual cell counting. Needless to say that this process is really time-consuming and the posterior data processing can be quite slow and error-prone. Which usually involves manual annotations of the anatomical regions of interest and sometimes even manual cell counting. Needless to say that this process is really time-consuming and the posterior data processing can be quite slow and error-prone. Therefore, we decided to automate our image analysis workflow by training several AI models using the Aiphoria platform, and we also developed tailored codes in Python to process the huge amounts of data output automatically and also to perform quality control of the data obtained. But you may be wondering, what is Aiphoria? Well, it's a cloud-based software that provides a platform to train AI models, which are based on supervised convolutional neural networks for fast and unbiased image analysis. I have to say that it's really user-friendly and it does not require any previous knowledge on AI to use it. So now maybe let's see a little bit how we can build a model using Aiphoria to try to set up a more automatic image analysis workflow. So the first thing that we would have to do would be, when we have decided which type of analysis we would like the AI to perform, we would have to upload representative images to the Aiphoria platform for training. When we have these images uploaded, we have to build a layer tree. And what the layer tree means is basically the architecture of the AI model. So basically, which features would help the AI model to recognize and to be able to end up performing the analysis we want it to. By this, I mean, for example, the usual hierarchy that we use is the one that you can see here. So we usually build the first layer, which is just able to discern what is brain tissue from what it would be a glass slides background. After, we, for example, commonly build a layer that is able to differentiate and to be able to identify one or multiple different brain regions in a mouse brain. And we can even make this even more complex. We can add more layers. Like, for example, if we want that the AI is able to detect a specific type of a cell or inclusion, we can also make a layer for that. So what I would mean with having to then decide which features need to be included in each layer, then we would have to annotate these features for the AI to learn from. And what I mean by annotating is literally manually drawing on the images what the AI should recognize as positive and leaving unlabeled what the AI should recognize as negative. When we have enough annotations and examples in different images, we can then run a training of a model. This all happens in the background in the Aiphoria platform. And what we are able to see afterwards is we're able to test this model and seeing how it performs on the images we had performed annotations on, but we should also see how it performs on images that we did not annotate and that were not included in the training. This is really important because at the end of the day, what we want is the AI model to be able to infer and extrapolate what it learned in the training images to be able to detect the same things, but in different and new images. So usually what happens is that on the first round, after seeing how the AI has performed in these trained and untrained images, we start detecting where the AI is having more trouble, which type of maybe features the AI is missing, or it's making more mistakes. So then this kind of guides us into making better or more annotations of a specific type. And of course, this needs to be done and checked for each layer that is included in a model. As you can imagine, we have to end up going through several cycles of these steps, but hopefully we arrive at a point where we are happy with the performance of the model on untrained images. And so then the Aiphoria platform allows us to fine-tune the performance of a model by being able to fine-tune some more advanced parameters. And so then we're ready to release our model. However, we can't really stop here. Before actually starting to use a model, I think it's really recommended to validate it. And this is really important because you can't just assume that the AI is now going to be doing perfectly. I think it's really important that there is a validation against like human researchers that are different from the training team, let's say. And also I think it's relevant to see how the AI compares and performs against maybe the previous method that you had been using for doing the same type of analysis. And I will come back to this later on in the talk. But first, let's see, for example, how we trained our most simple model, the striatal dopaminergic terminal detector, which was made to automatically measure the intensity of tyrosine hydroxylase or TH staining in the striatum in a whole slide image containing mouse coronal sections, which you can see some examples here in this slide. We first uploaded representative TH-stained images to iForia from separate experiments done by different researchers. And then I manually annotated the features that we wanted the AI model to recognize. These manual annotations are usually referred to as the ground truth, because this is what we will tell the AI, this is the truth that you need to learn from. And so in this case, for this model, the first layer that was trained was to detect the brain tissue. And I hope you can appreciate that there is a bright, thick white line around the full section in the left panel. And for the second layer, we built it to be able to detect the dorsal striatal regions within tissue sections. And what we did was then annotate many different sections and annotate the striatum. So hopefully you can see that there is a pale pink outline surrounding the dorsal striatum region. So after many annotations and several rounds of training, we ended up with the SDTD model. So as you can see here, what the model does, it first runs and detects the tissue from the background. So as you can see now, all the tissue is now a bit kind of like overlaid in white or grayish. And then within the tissue detected, the second layer runs to find the dorsal striatal regions. And so actually what we get in the output from this model would be the intensity of each the intensity of the staining of each of these separate striatal regions that have been detected. So before we jump into the actual validation of the SDTD model and also how it works in a real experimental case, I wanted to do a quick recap of the usual measures used to assess the performance of an AI model. The precision gives insight into how many positive predictions are made compared to the whole number of predictions the AI makes. The sensitivity informs us about how many positive predictions were detected out of all expected positive elements. And finally, the measure that is used the most in this field is the F1 score, which is the harmonic mean of both precision and sensitivity. And it is used to evaluate the predictive performance of the model, giving importance to both missed positive elements, but also to incorrectly detected elements. So the validation of the SDTD model, in our case, it featured 3 researchers who were not involved in the training, and they were asked to annotate multiple regions, which were then compared to the model's results for each layer. So you can see here in the top 3 images, it's how the 3 different researchers decided to draw the dorsal striatum. And then in the bottom, you can see how the AI model, the SDTD, performed. So these graphs that you can see show the F1 score of the model against the human validator against the 3 human validators in purple, and in gray it's the F1 score of the human validators against each other. So what we are doing is we are comparing the discrepancy of results of the AI against human validators to the discrepancy of the annotations between these same validators. And I hope, as you can see, for both layers, there is very high F1 score, meaning that the model's predictions are very close to the validator's annotations, and also as variable as the intervalidator variability, which is very good news. Now let's see a bit more of the application of the model. So you can see here in the right an example. So being able to put a numerical value to the biological effect of having a viral vector injected in the substantia nigra of mice unilaterally. And what you can see is how in the injected site, we are losing the intensity of the staining. And so what we then ask the Python code to do is to compute the percentage loss of the TH terminal intensity compared to the contralateral intact site. In this graph, you can actually see this for a group of mice who were controlled. So they were injected in the substantia nigra, but with saline, and also a group of mice that were injected with this viral vector overexpressing alpha-synuclein. The bar in gray is actually showing our previous method to do this type of analysis. So it was done through manual annotations in ImageJ. And post-processing in Excel, and we are comparing it in purple to the SDTD model. And what you can see is that, well, both methods are able to detect the same amount of loss in the alpha-synuclein overexpressing mice, whereas there is no loss to be detected in the control mice. And both methods are finding the same amount of TH terminal loss. Here in this graph, I'm showing you the correlation of both methods for each of the data points, so for each of the animals. And as you can see, there's a very high correlation coefficient of over 0.8. So maybe you are not so impressed with our most basic SDTD model. So let me show you what we built to assess neurodegeneration of the dopaminergic neurons in the substantia nigra. For this type of analysis, we built the nigral dopaminergic cell detector, or NDCD in short. And to train this model, what we did was we fed the Euphoria platform with mouse coronal sections stained for TH, but also counterstained with Cressyl violet or Nisl to help the AI be able to detect the neurons more easily. So the layer hierarchy or layer tree that we built for this model is the following. We first have a layer that detects the tissue from the background. Subsequently, the substantia nigra regions are detected within the tissue. And last but not least, we detect all the TH positive cells within the substantia nigra regions in a slide. Here you can see a bit more in detail how the model performs to detect these cells. And I wanted to mention that for the cell detection to be much more accurate and be able to discriminate between overlapping objects, we are using instance segmentation, which actually allows us to get very accurate morphological metrics per cell. The validation of this model ended up giving us this F1 score for the tissue layer, as you can see, very close to 100% for both the AI against the humans, but also between the human variability. For the brain region, we see a drop in the F1 score. So it seems like the AI is having more trouble recognizing the substantia nigra, but actually we see that there is no significant difference when compared to the intervariability of the human researchers annotating the substantia nigra. So that means that it's just anyone would find this a bit more subjective, and there would be just more complication in identifying, or more discrepancy in identifying this layer. And the F1 score for the TH cell detector is, again, really over 95%, meaning that we can be quite confident that the model is performing quite well compared to a human. So now we can see how the NDCD model compares to our most used method here in the lab. So quantification of neurons in the substantia nigra, we usually do it through stereology. And so here what is plotted is this loss of TH positive cells in the substantia nigra of, again, mice that were injected with saline, so the controls, and mice that were injected with viral vector in the substantia nigra expressing alpha-synuclein. And As you can see, there's more or less around 50% loss of neurons in the alpha-synuclein overexpressing mice, where the loss is around 0% for the controls. But also, there is no significant difference between the detection using both methods. And maybe a little bit more positive for the NDCD model, we see that there's also less variability using the NDCD model when comparing with the different samples. In case someone is curious about how actually the number of cells is calculated, here is plotted the actual numbers that were detected in the injected substantia nigra for, again, the same groups. And as you can see, there's no significant difference between both methods, and actually both methods are equally good at detecting a difference between a control group and the group with the lesion, or the overexpressing group. Here you can see again a correlation of the two methods with, again, a very high correlation coefficient for all these data points. So we have actually trained some more complex models to assess other neuropathological markers commonly used in preclinical PD mouse research. However, today I don't have time to go into them. But maybe the main feature that I wanted to highlight of these other models is that we have built in a very extensive brain region layer capable of detecting many different brain regions. So I'm going to show you just an example of how this brain region was trained for our microglial activation detector. So this is a model that we actually adapted because we transferred from an already existing model that was published by Stedzik et al. In 2022. And so what we did was we obtained this model from them, and then we built in an extra layer, so this brain region layer that we trained to detect many different brain regions across the whole mouse brain. So as a take-home message, I would say I hope I have convinced you that using these novel AI tools can be really helpful for a neuroscience lab. It's allowing us to have some less variability compared to our traditional methods. It also allows us to design more high throughput experiments. It's more quantitative in some ways, more unbiased, efficient. And it's also allowing us to compare much more between experiments from different researchers because we are using the exact same analysis pipeline. So last but not least, I wanted to thank, of course, the team that has helped me in this work. And I was also, if there are any questions, I would be happy to answer them. So thank you very much.